Hello everyone. So this is the second objectives, or we can say that the second topic which related with SARS-CoV-2 vaccine development. The first one I just get a brief information how to do by Moreland. Okay, next I will also, but okay, here I'm just focusing on SARS-CoV-2 vaccine development, present and future state. So this is the evolution, how it comes from. So I just go through beta coronavirus, and from beta corona, different type of viruses will form, and these are the SARS-CoV and MERS-CoV and SARS-CoV-2. These three are the COVID virus. So that the different approach to contain SARS-CoV-2, direct infection and indirect infection, some droplets, and the fecal oral, that's a matter of debate. Yeah, but we know that these are the different type of approach by which the COVID going to pandemic, and even still is in the matter of debate. So different vaccine is also in need, and that's why we are focusing about that, what the objective and what are the what are the scientists doing work, and how to just prepare vaccine. So this is the work related with this one and this presentation. And because I'm just going in more focus, so that's why coronavirus a brief information, that is a severe acute respiratory syndrome. So it comes from the respiratory tract, as everyone knows, and it is the RNA virus, and different type of SARS-CoV, MERS-CoV, and SARS-CoV-2. And SARS-CoV-2 is also called coronavirus 2 or COVID-19. And is RNA virus because RNA to protein. So it is a very time-saving process. That's why RNA to proteins. And so because someone is the DNA to RNA to process, so it time consuming. So time saving, usually virus have to just go through and make RNA as a genome. So RNA to protein. And also ultimately these SARS-CoV-2 have a protein, and they have some structure protein like envelope protein, membrane protein, spike protein. And spike protein having the S1 subunit and S2 subunit, and S1 subunit is a conformation state, 2 state, and open state and closed state. So I will describe a brief in later slide. So this is the coronavirus. This is a nucleoprotein, as you saw that, but nucleoprotein found on the on the interior, and which a protein and are associated with the RNA genome. But the surface protein is surface exposed spike protein and membrane protein and milk protein, different type of surface protein, because the protein which are found on the surface. And if you prepare some antibody or some type of, so it can be possible to interact with that surface. So that's why the person or most of the scientists have to work on the surface exposed protein, or called structural proteins. So person also have one of membrane protein, envelope protein, but usually spike protein here in this one. Right upper side is a spike protein up orientation, which is shown red in color, and it comes and bind with a ACE2 receptor. ACE2, that's one is a human cell, and usually also the human ACE2 receptor or HACE2 receptor. So ultimately it comes and bind, and by the process it just penetrate in the cell. So that's why we can say that the spike protein are the major important thing, because it is responsible to come and bind with the human ACE2 receptor, and after that the SARS-CoV-2 interact with the cell or penetrate to the cell. Okay, and this was the spike protein a pre-fusion state. Means suppose a spike protein of the COVID is in two state, pre-fusion state and post-fusion state. I will just go through in the next slide. So I will describe a brief information about pre-fusion and post-fusion. But here pre-fusion state in monomeric and trimeric state. This monomeric state interact with three monomer interact with this, so it make a trimeric, and the trimeric state are in the responsible to come and bind with human H2, and then SARS-CoV-2 enter in the cell. And this is the overview of, we can say, line diagram of the spike glycoprotein SARS-CoV-2. So signal sequence and terminal domain, receptor binding domain, and then S1, S2 site, and others are also, because we are just focusing on the spike S1 protein. So I'm just consider the NTD and RBD domain. So these are the two domain, and terminal domain and receptor binding domain. And this receptor binding domain, which is here from 331 to 528, and itself receptor binding domain itself contain receptor binding motif, which is the region which comes and bind with human H2 receptor. and that region from 438 to 506. Okay. Now the person usually confuses what is the up orientation, down orientation, open state, closed state. RBD, receptor binding domain in up orientation or down. Suppose if this spike and RBD in the here, so if RBD is up, so spike glycoprotein whole is called open state. Actually, there are three RBD. Suppose 1, 2, and 3 RBD. If one RBD up, or 2 RBD up, or 3 RBD up, in every case, RBD, if any RBD is up, so spike glycoprotein itself is called as open state. And if all three RBD is down, so this state is called closed state. Open state is closed state is a spike glycoprotein state, and up orientation and down orientation is RBD state. So this one is that this is the structure about what are the spike protein and such a 2-dimensional, 3-dimensional structure. And spike protein is a S1 and S2 domain, and S1 as seen here, S1 comes to bind to ACE2 receptor, I think, and this one is the ACE2 receptor, and this is a TMPRR SS2, that is a protease which assist S1 to bind with human ACE2 receptor. Okay, this is the important thing as I try to focus because usually what happen usually DNA to RNA, RNA to protein. Here DNA is absent, but ultimately I'm saying that what is the DNA to RNA and RNA to protein. So that is from transcription and translation. But what exactly happen in this prokaryotic cell or small cell, because they are just very fast growing, very replicating very fast, so they need protein in a very fast manner. So what exactly, as the mRNA come, the mRNA is not totally transcribed, even its mRNA start from translation. So mRNA is even coming from the DNA, and the mRNA is also going to the ribosome and comes to protein. So both are the co-translation process, understand? But here in the case, I'm just saying that here in this case, what happened is the 27 to 305 is the N-terminal domain and is a globular protein. 321 to 590 is receptor binding domain, is also a globular protein. So what exactly happened, because these are the, because N-terminal domain comes from, that is Vibgyor region, is VIBGYOR, V for violet. Okay, the same thing, blue to red. So these 27 to 305 region comes first, and 71 to 1144 comes later on, or in the last. So this globular protein 27 to 305, this globular protein comes first, and it hang on the cytosol. Understand? It hang on the cytosol, and then receptor binding domain is also come and hang on the cytosol. So both globular proteins are hang on the cytoplasm, and after that 696 to 711 there is a very large loop. So these two globular protein hang on the cytosol and a very large loop, and then 712 to 1144, these are the structural protein, and which is a helical region, so highly stable. So ultimately I am trying to say that these structure, receptor binding domain and N-terminal domain, are not directly involved to stability concept with the helical region. So if you would like to receptor binding domain as a vaccine candidate, you can use, because these domain are not directly interact with these things. But ultimately it would be better that have to create whole spike glycoprotein, because definitely some mutation may changes some other structural changes. So I would recommend that you have to use whole protein for the vaccine content, but yeah, it may have possibility to use from 27 to 696, and you can use this structure as a vaccine content. It can possible with as I think so. Okay, this is the mechanism. This is suppose the receptor binding domain is a spike glycoprotein, and this red box is RBD in up orientation. When it up, it bind with human ACE2 receptor and human ACE2. So after that, the spike protein which are on the surface of this COVID virus, and when it comes, suppose this is the COVID virus and this is spike protein, and when it is spike protein and this is S1 domain, it comes receptor binding, it opens receptor binding domain, and now it binds with human ACE2 receptor. Okay, after that it is become stable, because after that, because SARS-CoV-2 not a single virus, different how many millions of the virus are just going through respiratory tract. And when the spike of the COVID comes in the open state and it binds with human tissue, it becomes stable. When it's stable, then the human surface TMPR SS2 receptor, it comes and bind with S1 S2 region, S1/S2 region, then it cut it here, S1 in the second box, and S1 now liberate out, and now the rest of the region make some rearrangement and bind with the host cell. Understand? So ultimately what exactly happened? Suppose the first one, suppose the respiratory tract 100 billions of the virus just going through, and when it in the proper condition, this suppose COVID, then is a spike and a receptor binding domain. When it open, it bind with human H2 receptor. Now it becomes stable, and TMPRS2 receptor comes and cut the S1 domain, S1 liberate out, and the rest of the region, S2 region, it just make reshuffle and now bind with human cell surface. So ultimately spike and S1 because liberate out, and S2, suppose this is a human cell surface and this is S2 because S1 already liberate out and S2 region, and this is a COVID virus. Now after that, this is the spike S2 region, this is the human H2 receptor, sorry, human cell surface, and this is the this region is called S2 receptor, S2 region, and this is the COVID. So now when it come in bind, after that this just move like this because it just bind with this region. I suppose if you like a string and just like this, so it already bind with some portion, and now after that COVID comes in direct or indirect contact, and ultimately when it come into the surface, just go to some endocytosis and it come to the cell. So that's a mechanism, and it's important that you all have to get information what exactly we are looking for, because that's a basis to do some changes and some mutation I will describe in the later stage. So this is the crystal structure and what are the things. Yeah, so different vaccine candidate and different vaccine technologies have been used, but ultimately person have to work on the spike protein, either RNA or DNA or viral vector, some inactivated and live attenuated. Okay, these are also way, but the person have to work on the spike protein or subunit based vaccine or such type things. These are the top 10 vaccine, some mRNA based, some recombinant vaccine based, some inactivated, some activated. There are different ways, but yeah, different ways as I shown in, and I think I have taken this information from the WHO side, but ultimately my experience is the protein subunit, so definitely I'm just going through the protein unit. And okay, there are different advantages and challenges, but personally I prefer that is the third one is a no live component, so no risk of the vaccine triggering diseases. Such type of things is very important, because if you are just going to work on the virus, so no live component are important, and other thing is protein is highly stable. So that's third and fourth point are particularly my experience is more important, and rest other things are also challenges and advantages are important for. Okay, in the some previous slide I said the pre-fusion post-fusion. So this is the pre-fusion post-fusion state. What is a trimeric, then monomeric, then trimeric and monomeric. I will describe in the next slide how it will form. Yeah, this one. So this the cartoon region, this cartoon region is called S1 region, and the no, sorry, the surface region is the S1 region, and the S2 region is the cartoon region. And when I just third one, I just make a rainbow color, and ultimately I saw this red arrow is just going up and the cyan arrow is going up. So it shows that the pre-fusion state convert into post-fusion state. So pre-fusion and post-fusion both are the similar region, but only the thing is that suppose this is a pre-fusion state, and here some bondage, and when it comes in the some bondage and changes have some S1 S2 region, and now is now the post-fusion state. So the same thing. So that's important thing, because both have the similar sequences but different structure. But the thing is that we or everyone use SARS-CoV spike protein in a pre-fusion state, as a vaccine candidate, because in this state protein is not bind with human ACE2 receptor. So that's why the person have to work on the person want to work, or person want are recommended as a vaccine candidate pre-fusion state, but is highly unstable. Because if you use this sequence and just go through cloning all these things, and if you are looking for the protein should be look like a post fusion state, because post fusion state is stable, and if we both have the same sequence, so you are looking for the pre-fusion state, but the protein expressed in the post fusion, that's a problem. So that's why the person is thinking that you have to do some mutations, some changes, and arrest spike glycoprotein in pre-fusion state, which is highly stable state. You have to some sequences and just express this sequence and the protein. If you have a normal wild protein, and if you have to just go through and express, the protein should be expressed in the post-fusion state, because it's a highly stable. But if you have this sequence and you have to some changes, but changes are the many concept, and ultimately the antigenic region or epitope region are not changes concerning this in the mind, and ultimately you have to make some mutation and arrest the spike protein in a post-fusion state. So that's the objective. I think I just try and explain in a very possible manner. So this is Jackson Macron work, and he also has done the same thing, or how can I say he also, but I just read this article and I just try to do this work from my own. So this is the proline substitution and some salt bridge interaction, some field activity, and okay, this is the hidden one, but this work is published in the Nature article 2020. So this type of mutation he has done, and I also want to do this mutation, but some other mutation. So I just go through conserve server, what are the variable region, what are the conserve region, and some molecular dynamics, some normal mode analysis, and ultimately I found that the which region are flexible, such type of things. And so I just also try to do some mutation and arrest furin cleavage site, insert some disulfide, some hydrophobic interaction, insert some polar residue to enhance hydrogen, and some proline pricing to increase lubricity. And such type of things I had done, and I filed a patent. And so what is the strategy I had done? These are the some mutations which I think so just is a very time taking process. And ultimately this is the second one, the right one. What is the entropy and different type of intermediate states, normal protein folding funnel concept. But we are just looking for a stable protein, so we have done some mutation and just go through some to and fro process and get try to get some stable spike glycoprotein in a pre-fusion state. And so this is the way by the introduced mutation to arrest spike protein in the pre-fusion state. That's the K986 and V987, and is mutated by the proline, and that is a Jason Macron work. So I'm just saying that this is the work how to, I just try to do these things, but concerning similar concept. So these are some energy different type approach you just go through, and I just try to do these things. If I have some where I just looking for homology modeling, some threading, some energy minimization, such type of things. So this is the flowchart I just try to, and some by structure validation by process, and before and after I just get that okay, this is a energy minimization, such type of things, and just try to overcome the problem. And ultimately the this is my last slide. And so ultimately why I just say that what is the gist is the spike protein of the COVID have today mutated some things and arrest some mutation, and just some bioinformatics analysis just try to get information that protein should be expressed in the pre-fusion state, and this are mutated spike protein or spike genes, just clone and then cloning and then some animal stream. So patent are done a filing process. So that's the work I had done, and I hope I just explained best of my ease. And on the behalf, I'm highly thankful for your kind listening as such type thing, and I'm very thankful to do some collaborative work, and just please text me. Thank you very much.